Good day, everyone. This is Krija Oresco, and today we're going to be learning about lactic acid bacteria and their fermentation products. Now, kindly pause and read the objectives of this lecture. Next, what is fermentation? Fermentation is a word with many meanings. Pasteur used it to describe life in the absence of oxygen, while a more formal biochemical definition of fermentation is the process that bacteria use to make energy from carbohydrates in absence of oxygen. In common usage, fermentation also describes any biological process, whether oxygen is present or not. However, with a few exceptions, food fermentations are bioprocesses that change food properties, while the bacteria generate energy in the absence of oxygen. Fermentations add value to food by producing flavor compounds and carbonation, altering texture and increasing nutrient bioavailability. So food fermentation are bioprocesses that change food properties while the bacteria generate energy in the absence of oxygen, as I mentioned earlier. Next, the biochemical foundation of food fermentation. Um, we know in chemistry that energy is released when a compound is oxidized, meaning an electron is lost, as you can see in this slide. When one compound is oxidized and loses an electron, that electron must go somewhere. This electron goes to an electron acceptor, which becomes reduced. So, oxygen is the electron acceptor in the oxidation of sugars. However, in fermented food, there is no oxygen to serve as an electron acceptor. So, part of the sugar must serve as the electron acceptor. Catabolic pathways. The first pathway is the Embden-Meyerhof pathway. In the Embden-Meyerhof pathway, the glucose will be transformed into pyruvic acid and is the most important catabolic pathway. Fermentative organisms oxidize pyruvate to compounds such as lactic acid or ethanol. If its only product is lactic acid, organisms that use it are called homolactic. The next pathway is the Entner-Dodorov pathway. It is important in dairy fermentation. Lactose or milk sugar is a disaccharide broken down to glucose and galactose during fermentation. The EMP pathway cannot metabolize the galactose portion of the lactose. The wasted galactose can be taken up and metabolized by organism using the Entner-Dodorov pathway. But this organism produce only one 3-carbon intermediate and therefore one ATP molecules, in contrast to the ATP yield of Embden-Meyerhof Parnas, which is 2. It produces 2 ATP molecules. Next, the heterofermentative pathway. The heterofermentative pathway allows organisms to use 5-carbon sugars or pentoses that other organisms cannot use. Bacteria in the genera Lactococcus and Pediococcus are homofermentative, producing only lactic acid. Some Lactobacillus species are homofermentative. Some are heterofermentative, and some can use both pathways. So what are the uses of fermentation? Fermentation is used to preserve vegetables and fruits. It also develops the characteristic sensory properties such as flavor, aroma, and texture. It also destroys naturally occurring toxins and undesirable components in raw materials. Furthermore, it improves digestibility, especially of some legumes. Moreover, it enriches products with desired microbial metabolites, such as lactic acid or amino acids, to create new products for new markets. Lastly, it also increases the dietary value of the food. Next, let's proceed to the genetics of lactic acid bacteria. Genes in lactic acid bacteria can be located on the chromosomes or on introns. Introns are the insertions to the chromosomes. It can also be found on transposons, which are the transposable elements which move from one location to another, or it can be located on the plasmid, that is the extra chromosomal pieces of DNA. So plasmids are of particular importance in lactococci, in which they encode characteristics essential for dairy fermentation, including lactose metabolism, proteinase activity, oligopeptide transport, bacteriophage resistance, exopolysaccharide production, and citric acid utilization. So dairy fermentations. Many microbes are used to make fermented milk products. Note that Lactococcus spp normally form chains in liquid media rather than clusters when formed or grown on solid media. Pediococcus, on the other hand, are often found as tetrads, while Euconostoc have a single division plane and form chains. So again, the main bacteria used for lactic acid production are the homofermentative lactic acid bacteria. Heterofermentative lactic acid bacteria contribute flavor compounds as described um, before. The homofermentative and heterofermentative bacteria are often paired to give the flavor and texture characteristic of a given cheese. So these are microorganisms involved in the manufacture of cheese and fermented milks. So you can pause and read this. Pasteurization kills most of the natural lab and other bacteria present in raw milk. This facilitates the addition of lab starter culture. Star- so as I mentioned, pasteurization kills most of the natural lab and other bacteria present in raw milk. This facilitates the addition of lab or starter culture. Starter cultures allow manufacturers to control the rate and extent of acid development in fermented food. This results in greater process control and a more consistent product. So here are the secondary microbiota that are added to some fermented products to influence flavor and texture. Leuconostoc species and strains of Lactococcus lactis subspecies lactis metabolize citric acid and produce aroma compounds and carbon dioxide in cultured buttermilk and cheeses such as Gouda, Edam, Blue, and Havarti. Next, the heterofermentative lactobacilli produce ethanol, carbon dioxide, and lactic acid in more exotic cultured milks such as kefir and cow maize. The Propionibacterium frudentreichi, subspecies Shermani, I'm not sure if that's pronounced right, is added to make Swiss type cheeses in Swiss to make Swiss type cheeses. Its scavenging metabolism converts lactic acid to propionic acid, acetic acid, and carbon dioxide. The carbon dioxide forms the ice or host in Swiss cheese. Then, Propionibacteria also ferment citric acid to glutamic acid, a natural flavor enhancer. The next is Penicillium roqueforti. It is a secondary culture for the production of blue cheese. Then Penicillium camemberti is used to make camembert cheese. Then Streptococcus thermophilus and Lactobacillus bulgaricus is used in yogurt. Then Lactobacillus acidophilus and Bifidobacterium is added due to their popularity as probiotics. So here is the generic scheme for making fermented dairy products. First, preparation of milk at farm. Milk free of antibiotics is cooled in bulk tanks to negative or less than 5 degrees Celsius in 2 hours. Then next is receipt and storage at plant. Milk quality is determined before refrigerated storage for up to 72 hours. Next is clarification and separation. Sediment is removed by centrifugation and fat content is standardized. Next is formulation. Other ingredients are blended in to form the mix. Next is heat treatment. The mix is heated at 85 to 95 degrees Celsius for 10 to 40 minutes to kill bacteria and improve the texture of the finished product. Next, homogenization. The mix is homogenized to stabilize the product. Next is inoculation and incubation. The mix is cooled, a starter culture is added at 0.5 to 5%, and the product is incubated until the pH drops to the target value. Next is cooling, formulation, and packing. So the coagulated products is cooled and other ingredients such as fruits, flavors, or seasoning are mixed in before packaging. Starter cultures are used for rapid acid production to decrease the rate of contamination, for the production of flavor compounds, for more consistent product, and for a greater process predictability. Next, production of aroma compounds. Lactic acid is the main end product in dairy fermentation. It contributes to the acid taste but not the aroma. The main aromas and flavors of fermented milks come from acetic acid, acetaldehyde, and diacetyl. In yogurt, these are formed by Streptococcus thermophilus and Lactobacillus delbrueckii, subspecies bulgaricus. I'm not sure if that's how you pronounce it. On the other hand, in buttermilk and some cheeses, Leuconostoc species and citrate utilizing Lactococcus lactis, subspecies lacti, are used to produce the aroma compounds. Proteolysis is required for the growth of many lactic acid bacteria. Proteolysis also generates flavors in ripened cheeses. The free amino acids and peptides produced by the extracellular proteases can have positive or negative effects in the cheese. A major negative effect of proteolysis is the bitterness. This is caused by the breakdown of casein into hydrophobic peptides that are 3 to 27 amino acids long. Next, vegetable fermentations. Vegetable fermentation involve complex microbiological, biochemical, chemical, and physical reactions. Fermentations are also influenced by many external factors. These are classified into four groups, which are technological factors, ingredients, raw material quality, native microbiota, or the organisms that are naturally associated with the vegetable. Next, what are the steps in a typical vegetable fermentation? First, you need to select vegetables that are sound, undamaged, uniformly sized, and at the proper ripeness. Next is to pre-treat, or for example by peeling, blanching, or cooking. Third is to place whole, pierced, shredded, or sliced vegetables in fermentation vessels. These can hold from 100 liters to 100 tons, and you can also add a starter culture. Next, fourth step is completely cover the vegetables with brine. Seal the fermentation vessels to exclude oxygen and ensure anaerobic conditions. Fifth, you should let the fermentation take its natural course. The fermentation time depends on temperature, the type of product, and the bacteria present. The last step is to distribute the final fermented product, fresh, unpackaged, packaged, or pasteurized. Next, what are the ingredients and additive used during fermentations? Salt is added to fermentations for many reasons. In sauerkraut, I'm not sure how you pronounce, in sauerkraut production, um, sauerkraut, this salt enhances fluid release from shredded cabbage. It also helps in it also helps create anaerobic conditions in fermentation vessels. In addition, salt has a selective effect on the vegetable's natural microbiota. Increasing amounts of salt favor the growth of lactic acid bacteria and inhibit undesirable bacteria and fungi. Heterofermentative lactic acid bacteria are favored by the low acid concentration, or 1%, and are greatly inhibited at 3%. Higher salt concentrations favors homofermentative species, accelerating the fermentation. Salt content below zero point 0.8 often results in undesirable fermentation as well as in soft sauerkraut. In addition, ascorbic acid is also used in sauerkraut production to prevent gray or brown discoloration. Citric acid or sulfur dioxide is also used for this purpose. Sorbic acid prevents the growth of yeast, molds, and other microbes. So let's proceed to sauerkraut fermentation. Fermentation, or the first stage of fermentation, starts as soon as chopped cabbage is placed into the vessel. Salt is added to an equilibrium concentration of 2%. Next, the cabbage is tightly packed to form a ball at the top of the vessel. To create an anaerobic environment, the cabbage is covered with a plastic sheet, which is then filled with water. Under the anaerobic condition, the number of strictly aerobic bacteria decreases, while facultatively anaerobic enterobacteria grow for the first 2 or 3 days. During this period, dissolved oxygen is consumed by the microorganism and by plant respiration. The formation of lactic, acetic, formic, and succinic acid lowers the pH. Carbon dioxide may then generate foam. The second stage of fermentation, in the second stage of fermentation, non-lab are suppressed by anaerobic conditions and are overgrown by the lactic acid bacteria. Cabbage typically contains 4 to 5% sugar with about half glucose and half fructose. The fermentation of these sugars to lactic acid is initiated by heterofermentative lab, namely Leuconostoc mesoteroides, followed by Lactobacillus brevis. This succession of microorganisms is complete after 3 to 6 days, and the lactic acid concentration increases to 1%. Next, the third stage of fermentation is dominated by homofermentative lab. Their growth is favored by the complete lack of oxygen, low pH, and high salt content. The homofermentative lab increased the total acid content to 1.5 to 2.0%. Most sauerkraut is pasteurized when it reaches a pH of 3.8 to 4.1. Then the fourth stage of fermentation is optional. For economic reason, sauerkraut may be stored in fermentation vessel for up to a year. During the storage fermentation, Lactobacillus brevis and some heterofermentative species metabolize the pentoses released by the breakdown of cell walls. The acid content may increase to 1 to 2.5%. Next, pickle fermentation. Pickle are made from cucumbers. Pickles made by direct acidification outsell those made by fermentation. Dill pickles provide the vegetable components sold in restaurants that feed the fast food nation. Next, commercial cucumber fermentations are produced outdoors in vessels that contain 8 to 10,000 gallons of product. Starter cultures are rarely used after the cucumbers are covered with brine. The vessel is sealed with a wooden headboard. Next, cucumbers contain glucose and fructose at about 1% each. These undergo a homofermentation, typically by the naturally occurring Lactobacillus plantarum. Like sauerkraut, pickles can be stored in their vessel for up to a year. Then sweet pickles. Sweet pickles are fermented in a similar fashion using small cucumbers. After the fermentation, the cucumbers are washed and then soaked in 25 to 30% sugar. Next, damage on large cucumbers can be caused by bloater, and bloaters are caused by the accumulation of carbon dioxide produced by cucumber respiration or excessive growth of heterofermentative Leuconostoc mesoteroides. Next is high fermentation temperature. Lastly, high concentration of dissolved carbon dioxide. So these are the damage caused on or damage or the causes of damage on large cucumbers. Next, meat fermentation. In meat fermentation, the natural microbiota of meat is gram-negative, aerobic putrefying spoilage bacteria. Fermentation by lab prevents spoilage and turns the raw meat into a totally different product. The most common types of fermented meats are dry and semi-dry fermented sausages. So the steps in the manufacture of fermented sausages starts with the reduction of the particle size of quality meat by grinding. Next is the addition of curing milk or curing salt, 3% glucose, sugar, spices, and starter culture. Steps in the manufacture of fermented sausages. So next is the steps in the manufacture of fermented sausages. First, you need to reduce the particle size of high quality milk or high quality meat by grinding. Next, add curing salts, 3% glucose as a fermentable sugar, spices, and starter culture. Next, blend the ingredients. Then followed by vacuum stuff the meat into the casing. After that, incubate the sausages or ripen the sausages. Next, heat to inactivate the starter culture and pathogens. And lastly, age or dry the sausages. So that's the final slide. I hope you learned something.